Look at these shots. Just an ordinary blockbuster about mutant superheroes. But let's imagine, what if, pretty soon, guys like this start strolling down our streets? What's the current state of genetic manipulation at the cutting edge of science? Will herds of resurrected mammoths or Siberian rhinos roam the Russian tundra? For example, there's the extinct Thylacine, or Tasmanian tiger. Its reputation backfired cruelly. Europeans who were settling the island of Tasmania once decided that this predator was killing their sheep. In reality, that wasn't the case, but the extermination of the species was relentless, and the last Thylacine died in a zoo in 1936. Many years passed, and it seemed impossible to bring an extinct species back. However, the famous geneticist George Church and his team planned to resurrect the extinct predator using clustered, regularly interspaced short palindromic repeats, genome editing technology. What are the chances of this project succeeding? And what other animal species do scientists plan to bring back? Who is conducting these crazy experiments and why? And why do some experts call them extremely dangerous? How much does it cost to clone a deceased pet? And will the clone be exactly the same? And most importantly, is such progress in genetic modification natural? Or are there those who are deliberately promoting it? Be sure to watch this big mental film video until the end, and you will get answers to these and other tricky questions. We promise it will be interesting. Let's go. Often, projects to resurrect extinct species serve an important ecological mission. With the extermination of the Thylacine, Tasmania lost its main predator, and the food chains were disrupted. Scientists hope bringing back the Thylacine will stabilize the island's ecosystem by controlling herbivores like wallabies and kangaroos. Most of the Thylacine's genome is already sequenced. The very idea of resurrecting this species appeared about 20 years ago, but all previous cloning attempts have failed. In addition, there were no people willing to invest money in such an unusual project. But now, everything is different. The company received over $3.5 million in donations and opened a special lab. The Thylacine is considered ideal for resurrection because it went extinct recently, and scientists have high-quality DNA from it. Moreover, the natural habitat of the predator monkey has also been preserved, including the animals that make up the basis of its diet. The authors of the project are honest and openly say that in the end, they will not get a marsupial wolf, but something that resembles it. Here's what they write in their work. Our goal is to get as close as possible genetically. We can't bring back the body of Tien, but we will try to construct something very similar. The difficulty also lies in the fact that a pair of animals is not enough to revive the population, so they plan to release 100 Tasmanian tigers into the wild. If you think about it, it sounds scary. Scientists are essentially creating a new, never-before-seen species, even if it looks like the Thylacine. Then they release a whole pack of these animals into the wild, outside the lab. Even if all of them wear special electronic collar trackers, there's no guarantee that such a collar won't be removed. Besides, offspring born in the wild won't be wearing trackers anymore. What other risks do you think could arise from releasing a man-made, scientist-created species into the wild? Write your thoughts in the comments. The specialists also presented the public with a simplified diagram describing the process of resurrecting the Thylacine. First, the animal's genome is read. This stage is almost complete, with only a few % left to decode. The next step is to find a genomic donor. For this purpose, geneticists chose the fat-tailed dunart. Correct the distortions, engineer. It is considered one of the closest living relatives of the Thylacine and has several advantages. It breeds easily in captivity, and its genome is reportedly already decoded. The next stage raises many ethical questions among critics. The mouse will become the genetic template. Its genome will undergo extensive editing, allowing scientists to turn mouse cells into Thylacine cells. It's also remarkable that no one on Earth, including the scientists themselves, has any idea what exactly will come out of this. In the end, it's quite possible that gene editing will even be applied to individuals of a species called the marsupial anteater, and the fetus, which scientists tentatively call a Thylacine embryo, will be carried by a marsupial mouse. It sounds like science fiction, but experts are already working on this technology. There's an interesting point here. Thylacine babies, like the offspring of other marsupials, are only slightly larger than a grain of rice. According to scientists' plans, even a tiny marsupial mouse will be able to carry and nurse them. Let's discuss the next species geneticists aim to revive. Passenger pigeons survived many climate changes and the extinction of megafauna. They lived in North America, and at one time their population numbered in the billions. However, after tasting their tender meat, settlers began hunting the birds on a large scale. Large-scale deforestation only made their situation worse, and eventually, in 1914, the last pigeon named Martha died. The resurrection of the species is being handled by a California research organization. They promised to release the first individual into the wild in 2025. A group of American scientists is working on recreating woolly mammoths after the extinction of these giants, as well as woolly rhinoceroses, ancient bison, and roe deer. The blooming mammoth steppes disappeared, where other large herbivores such as wild horses, musk oxen, and moose used to graze. Subtitles created by Dimitar Zok. Now, in the Russian north, where large animals once lived, there is a barren tundra. Sometimes the prospects are assessed even too boldly. According to scientists, populating the Arctic tundra with a mammoth population could slow down climate change, and restoring the former Arctic ecosystem could theoretically stop the thawing of permafrost and the release of greenhouse gases. Researchers believe that enough genetic material has been preserved to clone mammoths or to insert their genes into their closest relatives, such as the Asian elephant. The company plans to use the $15 million raised for much of the project's experiments. Who knows? Maybe we or our children will be able to see mammoths in real life, just as our distant ancestors once did. The project's technical documentation states the first embryos will be obtained in a few years. However, some experts are concerned that the consequences of resurrecting extinct animal species are unpredictable. Specialists also question whether mammoths can ever be fully restored. They believe that, despite the project's ambition, it will only produce a mammoth-elephant hybrid at best. Currently, geneticists from Russia, South Korea, and Japan are also working on experiments to clone the woolly mammoth. No positive results yet, but the researchers' persistence gives us hope for success. Interesting experiments were conducted to restore the population of the bucardo. The extinct Pyrenean mountain goat subspecies died out due to poaching, habitat loss, and competition with domestic hoofed animals. The last individual died in 2000, and by 2009, scientists attempted to create its clone. Success was likely because geneticists didn't need to spend much time finding the closest relatives. As surrogate mothers, females of two other subspecies of the Pyrenean ibex were used. Spanish biologists created more than 400 embryos and implanted them into the wombs of 57 goats. But then problems began. Only 7 females became pregnant, and only one was able to carry the offspring to term. Unfortunately, the kid died just a few minutes after birth. Public criticism forced the scientists to stop their work on reviving the bucardo. The optimism of researchers working on animal resurrection is not shared by everyone in the scientific community. Some doubt that this is even possible in principle, while others fear that if geneticists succeed, they will face serious ethical problems. Is it humane to create an animal whose biology is so little known, and who will decide whether to keep them in cages or release them into the wild? Let's do a thought experiment right now. We're going to show you some very unusual extinct animals, and imagine what would happen if each of these species were brought back by geneticists. Where would they be kept? In the wild, or in cages? Or maybe in a special zoo with extinct fossil animals that you could touch and see alive? If released into the wild, how would they impact current ecological niches and food chains, and how would they fit in? Here, for example, is such a creature. Helicoprion, one of the longest-living prehistoric creatures, is a shark-like fish that was distinguished by its spiral clusters of teeth. The length of this monster could reach up to 4 meters, and this carnivorous beauty was called Dunkleosteus. Thanks to its terrifying size, reaching up to 10 meters and a weight of almost 4 tons, it was the top predator of its time. Here is another museum specimen with the unusual jaw of Platybelodon, a genus of extinct proboscideans that lived during the Miocene epoch, about 15 million years ago. Platybelodon? They had 4 tusks in the upper jaw, which were shortened and directed obliquely forward and downward. In the elongated lower jaw, both flat front incisors were fused together and transformed into a kind of shovel. Even an ordinary centipede from a pet store often causes disgust, and a 2-meter-long one, if it had survived to our days, would have been a nightmare for many. Its scientific name is Arthropleura. This insect weighed about 100 kilograms. It was a native European, living on the territory of modern Germany, Poland, and Belgium about 300 million years ago. With 58 leg pairs, the insect could freely but slowly walk through ancient Carboniferous forests. Another giant insect from the Carboniferous period was the giant dragonfly, with a wingspan of up to 65 centimeters. Like modern dragonflies, it was a predator. Scientists named the little one Meganeura, and they wondered for a long time why such huge insects lived in the past and don't exist now. Their theory cites Earth's high atmospheric oxygen as the reason. Scientists also found another cause when they altered the pressure in air bubbles trapped in amber, fossilized tree resin. It turned out to be 8 atmospheres, and the oxygen content in these bubbles was 28%, while nowadays the share of oxygen is 21%. In Hydropolis, the underwater city built by Jacques-Yves Cousteau, the high pressure made experiment participants' wounds heal overnight and nearly stopped their facial hair growth. Maybe the human body was originally adapted for a different atmospheric pressure. This theory claims giant insects and dinosaurs over 100 tons could only exist in a higher pressure atmosphere. Now, remember Diego from Ice Age? Yes, that's the mammoth in the world of cats, Smilodon, or more precisely, an entire subfamily of saber-toothed cats, lived in both Americas and Eurasia. Besides their fangs, Smilodons differed from modern cats by having a more powerful but stocky build, with short muscular legs and a thick neck, which allowed them to hunt even young mammoths and their close relatives, the mastodons. The fangs often broke because of their length. That's why all saber-toothed cats, strangely enough, didn't bite their prey, but first tried to knock the victim down and then drive their teeth in with a movement of their head. And that's why they had another feature. They could open their jaws up to 120 degrees. The Phorus rhacos, a 3-meter-tall bird weighing up to 300 kilograms, resembled a predatory ostrich. It couldn't fly, but didn't need to. Long legs let it catch any prey. Computed tomography scan data of one of the Phorus rhacos species showed that the bird had good intelligence and revealed one hunting feature. The skull of this bird was very fragile in the longitudinal plane, and this means that the Phorus rhacos couldn't grip its prey and could only strike it with its beak from top to bottom, like an axe. And now, attention! Horned gopher. It might sound like a ridiculous phrase, but such an animal really did exist in what is now North America, at the junction of the Miocene and Pliocene about 5 to 10 million years ago. Its remains are found in the United States and Mexico. It was a large rodent, up to 30 centimeters long, with 2 noticeable horns on its head, and their purpose is still unknown. You can think about this mystery in the comments under the video. I think there will be several theories. And this is Danahius, a huge, ferocious boar weighing about a ton, and he was ferocious not only towards his prey, but also towards his own kind. On the excavated skull bones, numerous bite marks from their fellow creatures are found, just like in many other species of giant mammals. Danahius was doomed by the Miocene cooling, which caused forests in North America to give way to steppes, leading to the extinction of many herbivore species. Danahius's dire situation worsened when amphicyonids, or bear dogs, arrived from Eurasia via the land bridge at today's Bering Strait. Bear dogs were not only strong, but also much smarter than Danahius, and eventually drove it out. This monster is unlike anything you've seen. A sloth as big as an elephant. Is that possible? Absolutely. Megatheriids were a family of giant sloths that lived in South America for over 1 million years and went extinct just 8,000 years ago. Moving on its hind legs, it looked like a huge bear with a large tail that it constantly used to support itself on the ground. Although megatheriids were slow and clumsy, they were by no means defenseless. The enormous size of this animal alone made it difficult to attack, and its strong, fast-moving front legs with 17-centimeter claws could kill any predator of that time with a single blow. And what is this anyway? Even its very name translates from Greek as born of delusion, hallucigenia, and the name is well-deserved, because when archaeologists discovered this invertebrate creature about 1 centimeter long, they couldn't figure out which side was up or down, and thought that the spikes on its back were legs, and its legs were tentacles. Although discovered in 1977, it wasn't until 2015 that scientists determined which end of this creature was the front or back. Hallucigenia lived on the seafloor. What it ate is still not exactly known, but it is believed to have been a filter-feeding animal. And this is the ancestor of modern arthropods, which looked so unusual that archaeologists named it Anomalocaris, meaning strange shrimp. 2 long tentacles, coiled in spirals and located on its head, were used to catch prey, and the presence of 2 complex compound eyes on small stalks gave the animal a truly alien appearance. This is also a YouTube celebrity. Titanoboa was a prehistoric giant snake as big as a bus. A motorcycle like this could easily fit inside it. These were real monsters among snakes that ever lived on Earth, weighing about 1 ton and reaching up to 15 meters in length. But despite its size, Titanoboa was a shrimp compared to most dinosaurs. Now, let's talk about unicorns. Not those delicate creatures from fairy tales and movies, but the tough Siberian unicorns. This ancient relative of the rhinoceros, the Siberian unicorn, went extinct about 26,000 years ago, according to scientists. They may have been the original models for fairy tales and legends. Now, a bit about its relative, the woolly rhinoceros. Mammoths weren't the only large furry animals that lived on our planet. Woolly rhinoceroses also roamed the snowy landscapes, and the remains of these animals have been remarkably well preserved. Scientists are considering extracting their DNA and using a modern rhinoceros as an embryo surrogate. Actually, they have a similar plan in mind for the mammoth. What do you think? Would it be great to bring such creatures back to life? Dodo? This bird is known to many from the book Alice in Wonderland. It lived on the only island on Earth, Mauritius, which is located to the east of Madagascar. In 1592, it was discovered and described by a Dutch expedition, and by 1662, the dodo had almost disappeared. No ice age, no cataclysm. The thing is, these birds couldn't fly at all because they had no enemies in their natural habitat. The funny dodo became easy prey for sailors and soon disappeared completely. Later, they became the symbol of the Dural Wildlife Conservation, and you can even see a statue of the dodo at the entrance. The namesake wildlife park. Let's finish with the prehistoric monsters. I wonder which ones from this list you would put in your special zoo, which ones you would leave in the wild, and which ones you wouldn't want to bring back at all. Write in the comments. And now, let's move from thought experiments back to modern scientific experiments. An unusual experiment is being conducted in China, where the first cloned, genetically modified cow has given birth to a healthy calf. The mother of the newborn was created in 2012. A special gene was added to boost muscle fat layers. The thing is, China has a shortage of breeds that produce marbled beef, and most Chinese people can't afford to buy imported beef. Many experts are convinced that the success of Asian biologists shows that a similar technology could work for resurrecting extinct species as well. Many countries have banned animal cloning for food purposes, and the European Parliament has even approved a bill prohibiting the import of meat from cloned animals from other countries. Experts have noted higher mortality and various abnormalities in cloned animals' offspring. China is actually leading the world in this field. The successes of Chinese geneticists are recognized by scientists all over the world. For example, China ranks second after the United States in the number of scientific articles in the field of biomedical engineering. However, Chinese geneticists have been involved in several scandals, mainly due to breaches of widely accepted ethical standards. For example, in 2001, a Chinese scientist grew rabbit embryos in a laboratory using skin cell nuclei taken from a 7-year-old boy to create hybrids. It was possible to use the cells. There are chronic geneticists with monkey genomes, which helps. How did the genetic behavior of autistic disorder form? Geneticists from the Chinese Academy of Sciences modified the genes of crab-eating macaques to induce autistic behavior in them. This helped to better understand the nature of autistic disorder. Meanwhile, they already clone primates and provide scientists worldwide with 90 genes and 95 transgenic monkeys for experiments. Now, let's talk about our little brothers. How would you feel if your deceased pet, so to speak, reincarnated back into your family? Demand for resurrecting deceased pets rises annually. Geneticist Huan Wu-Seok, founder of one such company, stresses that clients don't get an exact replica of their pet. Clones only look the same, but their behavior will inevitably be different. The scientist has found interesting marketing strategies and tells his clients that clones have every chance to become a better version of themselves for their beloved owner. The cost of the service ranges from $100,000 to $150,000. The cloning process is lengthy and requires significant expenses. DNA must be provided within 5 days after the animal's death. Otherwise, cloning is impossible. Huan Wu-Seok was a real hero in Korea in the 2000s. He was the first to clone a dog, and the local media made him a star. However, in 2012, it turned out that his work on the successful cloning of human embryonic stem cells was falsified, and Huan had to leave science. Later, he was also accused of embezzling public funds and illegally purchasing donor eggs from women. Now, the scientist, whom many consider a fraudster, owns a highly profitable business in resurrecting pets. Huan himself believes that human cloning is immoral, but animal cloning brings only benefits. The prices for cloning pets in different countries in 2022 are approximately as follows. Cloning a cat costs $35,000 a dog, $50,000, and a horse, $85,000. Currently, 4 companies are engaged in full-scale pet cloning. These are projects in the United States of America, China, South Korea, and the United Kingdom. By the way, experts immediately point out that the clone will be of the same sex, but may look a little different. For example, the cloned animal may have a different coat color or eye color. Biologists say coat color is influenced by both genes and fetal development in the surrogate mother. This issue remains unsolved. Sadly, clones often die during gestation or in their first months. Seeing all this, it seems science is advancing steadily, and soon woolly rhinoceroses, mammoths, and other extinct monsters may roam our planet again. But there is also sobering information. In May, experts from Denmark, China, Germany, and the United States published a study arguing that recreating extinct animal species is impossible. The scientists studied the issue using the example of the McClere's hopping mouse, an animal that was endemic to Christmas Island in the Indian Ocean when the first settlers began to arrive there. Along with them came ordinary black rats, which brought a disease unfamiliar to the local mice. As a result, by 1903, the McClere's mouse species became extinct. Scientists decided to sequence the mouse's genome to resurrect the animals. This is a necessary stage of the work. Although DNA was well preserved, the species was very similar to those alive today. Geneticists could not recover nearly 5% of McClere's mouse genome. Critics of resurrection are convinced that the existing limitations make it almost impossible to restore the genome of extinct species, since the half-life of DNA is only 521 years. Even from recently extinct species, only fragments of DNA remain, which means it's impossible to fully reconstruct the sequence. That's why resurrecting, for example, dinosaurs is impossible, and restoring species that disappeared recently can only produce something similar to the extinct animals, but not an exact replica. Experts from Australia, New Zealand, and Canada have assessed the risks associated with resurrection. there. Research indicates that reviving these populations could cause many more species to go extinct. The recreation of extinct animals is also criticized because such projects are extremely expensive. Back in 2017, scientists proved that investments in programs to preserve endangered animals would save many more species than comparable funding for resurrection efforts. For example, New Zealand would need to lose 33 living species to bring back 11 extinct ones. All right. So things have become a bit clearer regarding the resurrection of extinct animals. But science isn't just working on bringing back extinct species. There are also ongoing genetic manipulations. Let's take a closer look at this. A chimera is what they call a creature made up of cells from two genetically different organisms. For centuries, they were considered a fantasy, a myth, the product of the wild imagination of ancient peoples. But now a human-animal hybrid has become a reality. Here are just a few examples from recent times. In 1997, American scientist Charles Vacanti grew a human ear on the back of a rat. Once the organ reached the right size, it was transplanted to a patient. Geneticists in California have used pigs as donors for 2 years. Now, with the help of genetic engineering, they create a situation where human organs, such as a liver or kidneys, grow inside a pig's body. In 2003, the Chinese woman Hui Zhenshen combined human cells with a rabbit egg cell. And in 2004, researchers at the Mayo Clinic Hospital in Minnesota produced pigs with human blood flowing through them. Last year, Chinese geneticists conducted one of the most scandalous experiments in history. They created a chimera, a human, they created a monkey. Incredibly, the experiment was successful. Human DNA took hold in the primate embryo, but they did not allow a human monkey to be born. The group of researchers deliberately interrupted the development process. The fact is, there are still many questions about how such an embryo would develop. To what extent will human cells with human DNA appear? Could they enter the brain? And might they create human consciousness? In such a hybrid organism, despite the dangers of genetic experiments, there are people who are willing to try the craziest ideas on themselves. The Australian artist Stell Ark transplanted a third ear onto himself. The man implanted the hearing organ into his arm. And this is Elizabeth Parrish. She is called the world's first genetically modified woman. In 2016, the 45-year-old woman underwent a course of gene therapy against aging. The therapy was aimed at eliminating two main effects of the aging process: shortening telomeres and endurance. Telomeres are chromosome segments that limit how often a cell can divide. While telomere length varies by person, people are born with telomeres 15 to 20,000 nucleotide pairs long and die with 5 to 7,000 pairs. Their length gradually decreases due to a process called the Hayflick limit. This is the number of cell divisions, approximately 50, after which the aging process begins in the cells. In 2015, before we started the therapy, blood was taken from Elizabeth for analysis. Elizabeth's leukocyte telomere length was 6.71 thousand nucleotide pairs. After therapy ended in 2016, her blood was retested. Elizabeth's leukocyte telomere length rose to 7.33 thousand pairs. Theoretically, this means that the blood leukocytes became about 10 years younger. Parrish had the procedure in Colombia because such experiments are banned in the United States. The results of the study were confirmed by two organizations: the Belgian non-profit organization Healthy Life Extension Company and the British Biogerontology Research Foundation. However, these results have not yet undergone peer review and seem more like a trial balloon launched into the public consciousness. Officially, in most countries today, genetic experiments on human embryos are either banned or strictly regulated. In 2019, the Chinese scientist He Jiankui was sentenced to prison for conducting an illegal experiment involving the birth of twins from genetically modified embryos. He added a human immunodeficiency virus resistance gene without prior clinical trials. The scientist was fined 3 million yuan, over 27 million rubles, for illegal human genetic experiments. But the main thing is that the Chinese authorities confirmed the birth of the world's first genetically modified people. What exactly did He Jiankui do? He and his colleagues selected several married couples in which the men were human immunodeficiency virus positive. Then they altered the DNA of embryos conceived through in vitro fertilization using the new gene editing method CRISPR-Cas9. They aimed to make the children immune to human immunodeficiency virus since their fathers were carriers. To do this, the scientists tried to disable a single gene in the embryos that is responsible for coding the protein, which allows human immunodeficiency virus to enter the cell. Case materials show 2 women became pregnant and had 3 girls with altered DNA due to these experiments. Due to privacy, the trial was closed, and the children's fate remains unknown. These are examples reported by the media. And what kind of experiments are conducted in secret laboratories owned by the military or megacorporations like Bayer is something ordinary people are not allowed to know. We can also look at the recent past. 60 years ago, Soviet Institute of Experimental Surgery pioneer Vladimir Demikov conducted a highly controversial transplant experiment. He transplanted one dog's head onto another's body, connecting vessels to the host dog's heart. The experiment was successful, and over the next 5 years, Demikov performed several more transplants. And if we delve into truly ancient history, we can find mentions of the Anunnaki in ancient Sumerian texts. They could fly, had great wisdom, brought knowledge and science to people, and conducted genetic experiments. The official dating of these texts is around 6000 before Christ, but what's important is not the dating, which may or may not be accurate, but the very fact of genetic manipulation in the past. Iraq, 1845. While excavating Nimrud, English archaeologist Austin Henry Layard discovered a unique black limestone obelisk carved with the achievements of King Salmanasar III. His military victories and received tributes were among the images. There are strange hybrid creatures present. Did these beings really exist here on Earth? Or are they simply mythical creatures like those found in fairy tales from all times and peoples? After all, mythological traditions around the world are filled to the brim with hybrids. The Greeks wrote about centaurs, half-human, half-horse beings. In Indian mythology, Nagas were often depicted as serpent-like creatures with human heads. The Egyptian god Horus had the body of a man and the head of a falcon. It seems there's nothing difficult about inventing a mythical creature by combining in your imagination the head of one living being with the body of another. But the previously mentioned examples from modern scientific practice show that, at least at the current technological level, all of this is quite real. Another question is how much scientists realize what they are doing. For example, the genetic catastrophe in Brazil with GMO mosquitoes once again showed that scientists still have no idea about the consequences and dangers posed by the GMO mutants they create. Under the roof of Rockefeller Foundations, the theory looked perfect on paper. Strains of male yellow fever mosquitoes taken from Cuba and Mexico were genetically edited to make it impossible for their offspring to survive. Oxitec, a British-American company, systematically released tens of millions of edited mosquitoes into the wild over 2 years. In Brazil, the idea was that the modified mosquitoes would mate with females of the same type, which carry infectious diseases, and in the process would kill them. A team of scientists from Yale University and several research institutes in Brazil monitored the progress of the experiment, and here is the result. After the initial stage of the experiment, the mosquito population noticeably decreased, but after about 18 months, it returned to its previous level. Moreover, some of the mosquitoes developed hybrid strength, meaning that a hybrid of a regular mosquito and a genetically modified one created a more resilient population than the one that existed before the intervention. Scientists created resilient super mosquitoes. Oxitec mosquitoes were developed using a highly controversial gene editing method known as gene drive. Funded by a Defense Advanced Research Projects Agency division, this method and clustered regularly interspaced short palindromic repeats, gene editing aim to rapidly spread genetic modifications through entire populations, such as mosquitoes or humans, in just a few generations. It is noteworthy that the Brazilian experiment was funded by the Bill and Melinda Gates Foundation. In June 2018, Oxitec announced the creation of a joint venture with the Gates Foundation to develop a new strain of self-limiting friendly mosquitoes to combat the species of mosquitoes that spread malaria in the Western Hemisphere. The results obtained in Brazil show that the experiment is a catastrophic failure, as the new strain turned out not to be self-limiting. The Gates Foundation and Bill Gates have been supporting the development of radical gene editing technology and gene drive technology for more than 10 years. It's not surprising, as Gates has long supported eugenics, population control, GMOs, and is a strong gene editing lobbyist. In an article published in the Journal of the New York Council on Foreign Relations, which is one of the largest Rockefeller public organizations, Gates welcomes gene editing technologies in general and clustered regularly interspaced short palindromic repeats in particular. In the article, Bill claims that clustered regularly interspaced short palindromic repeats and other gene editing methods should be used worldwide to meet the growing demand for food and improve disease prevention, especially malaria. It sounds very kind, but in reality, gene mutations and agriculture with chemical poisons are not meant to feed people, but to reduce their numbers. The United Nations Food and Agriculture Organization says feeding the world with organic products is possible. At the University of Michigan, they found that switching to organic agriculture globally could already allow more food per person per day than now. The global shift to organic technologies is entirely feasible and straightforward. However, this transition also causes global upheaval in chemical agriculture. This is deadly for corporations making chemicals, pesticides, and GMOs. These are the same giant companies with nearly unlimited financial power and global influence over politicians and scientists. And now, regarding not the animal world, but the plant world, how do you imagine mutant plants? Maybe like this or like that. In fact, you see products with mutant plants in the store every day, and you buy them. The food industry has long added GMOs to many products due to complex recipes and processes. For example, you hardly ever eat corn itself, but corn syrup, corn starch, and corn flour are found in a huge number of products, from yogurt to cookies. The same applies to soy-based products. Palm oil is often shown as scary on television and is harmful. But it's not the only thing that matters. After all, 70 popular brands of baby food contain GMOs, and about 30 coffee brands on the market are genetically modified. What might a skeptical viewer say? The dangers of GMOs are overstated. Let's figure it out, and start with the 1990s when a terrible epidemic hit Nicaragua, Mexico, and the Philippines, and scientists couldn't find the source for a long time. Completely healthy young women in their second or third trimester of pregnancy suffered miscarriages. Repeated attempts to conceive ended the same way, and then infertility followed. Doctors were at a loss. The only thing that united the victims of this strange disease, they had all participated in a mass vaccination campaign against tetanus, which was overseen by the World Health Organization and funded by the Rockefeller Foundation. It was hard to doubt the sincerity of their good intentions. However, the Roman Catholic group Comite Pro Vida in Mexico City tested the vaccine vials and found some alarming results. The solution contained human chorionic gonadotropin, which is a natural hormone necessary for maintaining pregnancy. But when combined with the tetanus pathogen in the vaccine, it led to the formation of antibodies against the natural hormone necessary for the fetus. In other words, the vaccine was a covert form of abortion. When the Rockefeller Foundation and World Health Organization conspiracy was exposed, they took a different path with GMOs, although the scenario was the same. The same things began occurring in Zimbabwe and Guinea as in Nicaragua, Mexico, and the Philippines. But this time, the affected women were united by their fondness for canned corn. Contrary to expectations, tests found no life-threatening components in the cans, and preservative levels were normal. The problem was with the corn, specifically its genetic modification. Antibodies were taken from women with a rare condition known as immune infertility. The genes that regulate the production of these infertility antibodies were isolated and, using genetic engineering methods, inserted into the genome of regular corn seeds, and those went to third world countries. Thus, Africans were unknowingly planting genetically modified killer corn with built-in hidden contraception in their fields. Genetic modification was discovered by the American corporation Monsanto in the mid-1980s. This is a major transnational company founded in the United States back in 1901. Now it has merged with Bayer. And we talked about the fact that currently 3 megacorporations control the world's food supply in our video about the Rockefeller Foundation. We highly recommend watching it if you haven't seen it, so you can get a more complete picture, according to Monsanto, which is closely connected to the Rockefeller Foundation. The main goal of creating GMOs is to increase crop yields, create new biological units, both plants and animals, and also to make crops resistant to harmful weeds. The slogan of all these cannibalistic corporations is simple: there are too many people in the world and too little food. And this, perhaps, is their main answer to the question of why humanity supposedly cannot live without GMOs. But is it really true that they want to feed us? Or maybe this feeding has other goals, especially since, historically, they have always worked on means of destroying people. From 1941 to 1942, Monsanto mainly produced military products. During World War II, Monsanto received about 2% of all budget funds allocated by the United States government for the needs of the military industry. For example, almost $6 million were allocated from the federal budget just for the production of chemical weapons. During the war with Vietnam, Americans sprayed Monsanto's Agent Orange to destroy tropical forests where partisans hid. This chemical is the precursor to modern herbicides and pesticides. The consequences of such spraying in Vietnam are still visible today. Children were born with defects, with undeveloped arms and legs. Those who were exposed to the spraying as adults still suffer from numerous purulent ulcers on their skin that do not heal. Returning to the topic of transcorporations wanting to feed everyone on the planet with GMOs, it's worth turning to analytics. In the United States, almost all agricultural products have been subjected to GMOs today. They have excellent corn and soybean harvests. Everyone should be satisfied and well-fed. But is that true? According to statistics, people from low-income families in America are more likely to suffer from cancer, diabetes, obesity, and swell up in the American zombie land. It's not from hunger at all, but from cheap mutant foods. Here's just one example. The lion's share of the food industry in the United States when it comes to oils is taken up by canola oil, a genetically modified product that is mainly produced in Canada. In fact, it's rapeseed oil. In 2009, 90% of the rapeseed grown in the United States was already genetically modified, and now the percentage is close to 100. It's cheap and rough, fine for the poor, but the rich won't eat it. And why Canada specifically? Because there's simply no regular rapeseed left there. Foreign genes can travel with pollen, turning ordinary plants in the fields into transgenic ones. Genetic contamination directly results from GMO production. One of the biggest scandals happened in 2006 with American rice from Bayer Crop Science, which spread from test fields all over the world. And the most significant consequence of genetic contamination was the complete and irreversible disappearance of non-GMO canola in Canada. But there is a place on the planet where, deep underground in the Arctic soil, a huge collection of seed samples from all over the world is stored, untouched by GMOs. And this place in Svalbard is truly grand. The construction of the Doomsday Vault began in 2006 under the auspices of the United Nations to preserve the seeds of all agricultural plants that exist on Earth. Biologists state that the number of crop varieties in the world has exceeded 2 million, with more than 100,000 subspecies of rice alone. Initially, this ambitious goal seemed unattainable. It appears that powerful people are committed and using all resources, making the seed vault's scale impressive. Who actually financed the project is not reliably known. The facility is located in Norway. The United Nations claims that this country funded the global project, and the initial investment from the government alone amounted to $9 million. But it's unlikely that the kingdom would have agreed to shoulder the burden alone, and we'll talk about that later. Completed in 2008, the facility's exterior resembles a bunker entrance. Each country has its own compartment in the global seed vault, with space for millions of crop varieties. Nations send samples as best they can. North Korea, for example, has handmade wooden boxes that stand out among the others. At the same time, 500 seeds are required to preserve each species. The global elites intend to store the necessary amount, and the maximum capacity of the cryogenic storage is over 2 billion seeds. Scientists spent a long time deciding where to locate the bunker, and after calculating the optimal conditions, they built it at a depth of 120 meters in the village of Longyear. When choosing the location, they even considered the low tectonic activity in the Svalbard archipelago area. Almost nothing happens here. With permafrost and rocks, it's impossible to find better conditions. In the cold and dry air, there are very few microbes, fungal spores, and dust, and the remains of animals and abandoned human structures remain unchanged for decades. The creators equipped the entrances with blast-resistant doors and airlock chambers. The reinforced concrete walls, 1 meter thick, can withstand a nuclear warhead strike or a major earthquake. To get deep inside the storage, you have to go through 5 doors with combination locks. The security is such that many can't get through. Only political leaders can dream of a bunker like this. Powerful refrigeration units run on local coal, which eliminates any problems with fuel supply for their operation. Permafrost also helps with this issue. Therefore, even if all the equipment completely fails, it would take several weeks before the temperature inside the storage rises, even by a couple of degrees. Scientists estimate the air inside won't reach 0 degrees for at least 200 years, and the climate features of northern Norway also help reduce the costs of maintaining the necessary temperature inside the facility. But who actually invested millions of dollars in this project? And is this really insurance against a global catastrophe? Or is it a tool for total control over the world's food chains? Be patient, and you'll learn everything. The vault's seeds are high above sea level, deep in the rock. The likelihood that it could be flooded by seawater or that the permafrost will melt in the next tens of thousands of years is almost zero. The seeds are stored in sealed envelopes, which are packed in special 4-layer bags. These, in turn, are placed in sealed containers that are kept on shelves. A steady minus 18 degrees Celsius slows metabolic activity, stopping seeds from quickly germinating or aging. If you think that boring scientists have focused only on cereals, you're mistaken. For example, the Irish government has supplied barley seeds for storage, which are needed to brew traditional Irish beer. So, if you ever want to relax and have a beer, after all humans are gone, and only ruins and ashes remain around you. Theoretically, there shouldn't be any problems with this, but of course, the seeds that are stored first are those of plants used for food. The very first 7,000 samples from 36 African countries were sent to Svalbard from the International Institute of Tropical Agriculture, based in Nigeria. In 2018, the number of storage units that had already been collected in the seed bank exceeded 1 million. Russia is transferring collections of food crops, such as wheat, barley, oats, and legumes for storage. Russian scientists see this reserve as the most valuable for the country during global catastrophes. Although the bunker is meant to help humanity in the event of the end of the world, that is not its only purpose. For example, if as a result of wars or natural disasters, countries lose their seeds, they can be replenished from the collection stored in the vault. The secured facility's doors are opened only a few times yearly to minimize seeds' exposure to the outside world. The party that submitted the samples for storage retains ownership of them. Norwegian authorities and bunker management cannot grant access to storage funds without approval from the party that submitted the samples. At least once, there has already been a need to retrieve seeds from Svalbard. Don't worry, you haven't missed the apocalypse. At that time, Syria was the one seeking help. Local researchers needed wheat and oat seeds that would be resistant to high temperatures. Syrian scientists requested the return of 130 seed boxes, and their request was approved. The shelf life of seeds depends on the specific plant species. For example, peas can retain their germination ability for 20 to 30 years, while sunflowers and some grains can last for many decades or even centuries without losing their ability to sprout. Some of the seeds are taken from the collection and planted in the soil. They will sprout and produce new seeds, which will replace the old ones. This is how infinite storage is achieved, but still, for what kind of future is this preservation being ensured? First of all, we need to look at who the sponsors of the Doomsday Seed Vault are. Sources mention the involvement of Bill and Melinda Gates' money. Among the investors is also the United States agribusiness giant DuPont, a monopolist in the creation of genetically modified seeds. The Rockefeller Foundation, which we cover in another episode, is also here. Previously, the foundation invested over $100 million in genetic engineering to support developing so-called pure plant and animal species. Until recently, Rockefeller's Green Revolution was part of a grand strategy for the development of genetic engineering of plants and animals. It was intended to concentrate control of food chains in corporate hands, taking away the ability of small farms to be independent. An important aspect of the so-called Green Revolution was the spread of genetically modified hybrid seeds in all countries. Their feature is the lack of reproduction, meaning that through genetic engineering, hybrid seeds are designed with protection against propagation, and after the harvest, the seeds obtained will no longer be viable. Thus, to achieve high yields, farmers have to buy seeds every year, and workers on the land can no longer produce viable seeds on their own. The annual purchase of commercial seed stock and necessary pesticides has already made small farms in the United States directly dependent on agribusiness, large companies, and fertilizer producers. It's also interesting that Bill Gates and Rockefeller, who are now investing huge sums in preserving seeds from a possible but not very likely end of the world, recently invested millions of dollars in the Alliance Project and the Green Revolution in Africa. The project's main goal was to promote genetically modified seed distribution. Former United Nations Secretary-General Kofi Annan, himself African, thanked the Rockefeller and Gates Foundations for supporting the African campaign at the Cape Town Economic Forum. Americans are very consistent and persistent in their desire to get the countries of this continent to use GMOs. Many projects include training African scientists in genetic engineering in the United States and are funded by the World Bank. The Seed Vault is under the trusteeship of the Global Crop Diversity Trust. The board of the Global Fund is headed by Margaret Catley Carlson, whom some researchers accuse of promoting birth control, sterilization, and population reduction programs in third world countries. Another corporation involved in the creation of the Global Seed Vault is the multinational company Monsanto, which is considered a world leader in plant biotechnology. Monsanto, together with the United States government, holds the rights to a technology in which seeds are capable of self-destruction after one harvest. Thus, this multinational giant holds a certain power over the supply of food to humanity. Still, many people see Gates and Rockefeller as generous benefactors, wealthy, kind individuals who donate to charity. One thing is clear: the companies that initially built their capital thanks to war, then continued to profit from producing chemicals for agriculture and now from genetic engineering, are clearly winning. And apparently, they have certain goals that go beyond just satisfying people's appetites. Did you know that, in addition to the Rockefeller Foundation, Rockefeller University also conducts research in transgenic microbiology? It's a strange university with no students, and it was founded in the same year as the Monsanto Corporation. Scientists at the university say their research will one day change everyone's lives. Who would doubt it? But what kind of changes are we in for? Supporters of GMOs claim that foreign inserts are completely broken down in the gastrointestinal tract of animals and humans. However, according to Russian geneticists, the consumption of organisms by one another may underlie horizontal transfer. Since it has been shown that DNA is not completely digested, and individual molecules can enter the cell and the nucleus from the intestine and then integrate into the chromosome, V.A. Gvozdev. As for plasmid rings, the circular form of DNA makes it more resistant to destruction. Genetically modified inserts have been found in intestinal microflora. During research conducted by a group of British geneticists led by Gary Gilbert, it was found that DNA from genetically modified food cells is taken up by bacteria in the human intestinal microflora. The capture of genes and genetically modified plasmids by the intestinal microflora was also noted in the work of other researchers. Transgenic inserts have been detected in human saliva and intestinal microflora, in the blood, and in the cells of various organs of the intestines, spleen, reproductive organs, heart, brain, skin, and other organs of mice and their offspring. According to the Center for Dairy Control in the southern German city of Weinsteffen, genetically modified inserts were found in the milk of cows that were fed genetically modified feed. Once, Ted Turner, the founder of Cable News Network, said, "We need to stabilize the Earth's population. We have too many people." The words on the Georgia Guidestones immediately come to mind. Let the Earth's population never exceed 500 million. While remaining in constant balance with nature, wisely regulate birth rates, increasing the value of life preparation and the diversity of humanity. Think about this, and the more you look for information on this topic, the better your puzzle will come together. Well, that's all for today. Thank you for liking the video, and see you in the next episodes on our channel. See you soon.